لهذا الملتقى، ولو سمحتم سنتكلم بالإنجليزية لأن أكثرية العناوين باللغة الإنجليزية، وهنالك عدد كبير من الحاضرين اللي يتقنوا اللغة، يفهموا اللغة هذه، بتقدر نسهلوا العملية على المترجمين. So we will start the session of today morning on camel biology and more specifically genetics, physiology and reproduction. The planning is including one key note or introductive conference and 5 oral presentation. And we will start with the keynote lecture, which will be given by Professor Abdelrazik Kakar, and my colleague will give a brief presentation of our colleague.
Merci. Bonjour tout le monde. Nous allons commencer cette session comme il a présenté M. Darghouth par la présentation de M. Aziz. Juste une short biographie pour M. Aziz. M. Aziz a travaillé en tant que directeur des opérations de la ferme d'élevage des chameaux El Ain depuis 10 ans. Il a également travaillé comme professeur et doyen de la faculté des sciences animales vétérinaires de l'Université d'agriculture de l'ESBA et des sciences de l'eau et de la mer au Pakistan. Également, elle a documenté des différentes races de chameaux au Pakistan, mettant l'accent sur la potentiel de la production laitière en particulier. Toute sa carrière articulée autour de la transformation du chameau en bête de somme en un animal de ferme durable. Elle a publié de nombreux articles scientifiques sur les différents aspects de la production de chameaux. Monsieur Razik a fondé également un forum de défense des chameaux Camel for Life International qui a promu de lait de chamel au niveau mondial avec un site web de pointe, Camel of All Info. Monsieur Razik, vous avez la parole. Donc, Mr. Razik, you have to speak. Thank you.
Thank you very much. Bismillahir Rahmanir Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Assalamu alaikum, and I am very happy I arrived here because I passed through very challenging time and I stayed in the airport for more than 7 hours. Our flight was delayed because of the political situation in the region. So I am very happy to be again in Tunisia. We were here in 2009 for Camel conference. So my talk will not be very, it is from the field, from the industry. I have brought some questions to discuss with you people, with the academics, with the researchers, with those who love camel or who are interested in the camel world, in the camel milk. So my, the topic is we should choose one, or what we think about the productivity or sustainability. What is the most important in the camel world, in the camel milk dairy? And then we will see what is important to shape the camel dairy industry in future, because you know the world is changing, the global warming is there are many challenges. So I am 100% sure that the future is the camel. So But what type of camel milk industry or dairy industry we want to establish in the future? That is the question which I have brought to you people. So, how it works? I don't know. Please help me. How to go to next? Next. Okay. Thank you. So we have some questions like the main thing is if you have policy, you can find the solution. If there is no policy, I mean, you are traveling or walking in tunnel. You don't know where to go. So the first thing, I mean, we will cover all the other issues, but the policy level talk is very important. We should know that what type of camel we want to see. Either we have to follow the cow model or what type of model we want to have in the future. So that is very important. And but you know the productivity, like productivity, what type of productivity? Productivity actually ensures the sustainability. If we will not produce milk, we will not get money in return. So there will be no money, there will be no business. That is very important. And then we have to address the demand because there is demand for the camel milk, especially in China. In China, there, I mean, if you will convert all the milk, all the camels into milk, then you cannot fulfill the demand for camel milk in China. So should we follow the demand curve? Or we have to generate, I mean, demand, I mean, to work on camel milk advocacy. And then, I mean, what are the intensification pros and cons? Because there are many farms now in UAE, in Saudi Arabia, now even in Kazakhstan, they are intensifying the camels in one farm. So what are the pros and cons? What will be the consequences? I mean, it is not just to happen. There will be consequences. And then we have to follow the quality, the volume or the quality, because we need more milk or we need the quality milk. That is the question. And then should we support the community-based dairy development in the camels? That is very important. Or we should only support the intensive farming system, I mean, to produce more and more and more. And then we have to think if we are going to produce milk for the camel, for the local market, or we have to export, because many countries, when you go there, we talk there, they are looking for Chinese market. They are not looking for their own people, I mean for their own local market. That is very important, where we are looking for. And then we have to make our own model. What type of model we want to have in the camel milk business, because we don't know. I mean, there are many players. Each player is working in his or her own area. We don't know which model we have to follow. Then we have the standardization issue. Dr. Bernard Fay is in front of me. They had like one very good discussion few days before. I am like, we should think on this issue again and again, what type of standards there should be. Because I'm producing milk. We are producing more than 7,000 liters daily. Daily we are producing 7,000 liters. But when we receive from the factory a message, they say your milk is less than 2.5% fat. I mean, it is like bomb for us. I mean, we are not the people to engineer the milk. The milk is synthesized by the camel, not by us. So if someone will impose standards on the camels, what will be the consequences? These are the questions. So, should we go to this model or we have to keep this model? These are true from the field, not fictions, not stories. This is the reality of the field. Some people get angry on me. They say, why I show this picture again and again? Because this I took in one Hungarian farm in Hungary, in Europe. I mean, you can see the cows. I mean, you can see the dirt. You can see the suffering the animal they face. But on the other hand, you can see in my farm, I can claim that I can sleep with my camels. There is no issue. There is no issue. That is neat, that is clean. So we have to think about the model, what model we are looking for. So I don't want to make you emotional, but these are the ground facts. I mean, the realities from the field. I am from the field. So then productivity ensures sustainability. Like I in brief, I said, if there will be milk, there will be income. There will be income, there will be farm. Otherwise, the farm will shut. So the camel, in my view, I have my own definition of the sustainability. If a herd of camel that can maintain minimal herd growth up to 3%, then you can sustain this farm. Otherwise, I mean, from inside, the story is that the camel herd growth is very low in the farm conditions. There are many reasons. So if there will be 3% growth over time, ensuring the profitability and posing the lowest eco footprint, because this is very important. If your farm is producing slurry, gases, emissions, and you induce a lot of antibiotic, chemicals, drugs, pest control, and these things, I mean, you will go to zero level. You have no, I mean, there will be consequences. So we have to avoid the consequences which are already happening in the dairy cow model. So then we have to, I mean, productivity or this 3% growth is impacted by the conception rate, by the calving rate, by the calving interval, mortality, culling, early calf mortality, because we are playing with these figures. Like, what should be the culling rate each year? I mean, 8%, 7%, 6%, how much we have to cull? Because there are many retired animals in your farm. Because they are either their teeth are blocked, there is like a lameness, they cannot walk properly, they are not reproductive, they cannot produce camels for you. So I mean, there are many factors which affect your policy for the development of the farm. Then we have like and then there is the major income. You know, in the farm, it comes like our farm. It comes from the milk. We don't have other source of income. So there be like we have to diversify the farm income. That will also help in productivity. So there will be productivity. There will be sustainability. So the other income can come, I mean, like we are getting some income from the male calves, from the culled animals. Additionally, we can generate income from the manure, because the manure is like the one camel is produced almost, you can imagine how much dry manure per day. It is almost 12 kilo. So if you have 1,000 camels, you have 1.2 ton of manure every day. So if we will generate, I mean, economy from this source, it will be an additional income to the farm. Then there is wool, there is tourism. I mean, many things can be do. Then there is value addition to the camel milk. There is misspell, not value, it is value addition. So if we will add value to the camel milk, I mean, we can generate some additional amount, some income. Like some people, they are working on camel milk cheese, some, I mean, like ice cream. We started ice cream, cheese, camel milk coffee, and many, many things. But ultimately, the train reached to the point that we have to still, I mean, export to China. So these are the things which are, I mean, positively or negatively affecting the productivity. And then you see, last year there was a conference in Dubai. This is World Environment, the COP28 on climate change. So the lube lubab, the extract of the meeting was the COP28, that we have to use, or we have to use the adaptation, the power of adaptation of animals and plant as a source of to ensure the food security. Because if they will be not adapted, they cannot survive in the climatic conditions, because the temperature is going up and up. So therefore, the camel is, now here is the question, I mean, in such situation, who will support, I mean, the other livestock? I mean, the camel is the only solution, especially in those regions which are hit by the climate change. The temperature is like, now 50 is very common in summer, very common. Before, like, there was like one day, 2 days, there was harsh weather, then the weather became little bit normal. But no, there is no normalization anymore in the climate. So we have to look for the livestock, for the plants, which are highly adapted to the local harsh environment. So you can see that this graph, unfortunately, sadly, this is going up and up. So 55, 51, 52, I mean, we have no other option except so the camel productivity is ensured in such climatic conditions without any inputs. I mean, you don't need to provide cooling system, sprinklers, a lot of water, many to know. The camel easily they survive in such conditions. Not only survive, but they are producing milk also. And the same, it is almost the same talk, the same thing, that they are highly adapted, I mean, to the high temperatures, I mean, they eat whatever the other animals do not go even close to those plants. Like this is Zygophilum catarensis. Now they have changed the name, the botanical name, Tetraina catarensis. I mean, no animal can go close to this. I mean, this is only the camel, they consume and they convert it into the milk, into the meat. So they are highly adapted to the desertification, to the aridity. They are, have very low pressure on the natural resources. They are not in competition with human to use cereals or something, and they can consume 8 to 10 times less water than cow in the same environment to produce 1 liter of milk. So these are the very important points which we should keep in our mind. Then there are like many references for the emission. There is low emission globe. You might have heard about the global agenda on sustainable livestock. They are always talking and speaking about the greenhouse and the slurry, humid dairy environment. The camel is very fit to the dry environment. There is like no slurry, no humidity, no dirt, nothing. Like I showed you in the picture, one can even sleep in the camel farm. So then the camel produce, like I told before, 12 kg of manure. So, and also the camel have less health challenges in the same environment. You have to induce a lot of antibiotics, a lot of drugs, a lot of things to the dairy cows in the same condition, but the camel need very lesser interventions for their health. And I will go a little bit quickly. There is adaptability, low water use, low inputs. They can digest anything. Even I have seen many times, I showed you in my videos, that the camel, they are eating the cartoons, the papers. I have seen myself. They can digest anything. And low gas emission and grazing, they do not graze intensively in small place. They go walk, eat, take one bite from one plant and go to the other. So then there is in last 10 years, there is not even 20 years, there is one big shift or change in the camel world. There is intensification in the camel farming. So what are the pros and the cons for the, I mean, for this shift? And what we can see, I mean, like the intensification, there is one, there are few benefits, so few positive points. Like you have homogeneity in the production. You have the milk all around the year. I mean, there is very lesser dent in the milk production. In the pastoral system, I mean, in some season there is no milk at all, and then we have like we have like very good value chain. You just produce milk and put it in the chilling tank and transfer to the factory. Within 2-3 hours, all the process is completed. So that is the benefit, and there are like. There is one point which we have noticed. I mean, this is our observation, that the camels which were born at our farm, I mean the heifers, bakarat in Arabic we say, they have better performance than the camels which we purchase from outside. So these are the three main pros of the intense dairy farming, camel farming. Then there is like, if you keep more camel in one place, they have health challenges, many ticks, many, I mean, other ticks born or the other pest-borne diseases, blood parasites. And also there is for that you have to use more drugs, more chemicals, more antibiotics, many things. So ultimately these things flow or drain to our food, I mean to the milk and the meat. And then we have the environmental issues. I mean, whatever, when you put more camel in one place, there are like more things. So, I mean, the khulasa kalam, we have to work on two-pronged, I mean, like here, I mean, I will talk a little bit about the demand of the camel. I mean, the camel milk, there are two main drivers behind the camel milk attraction. One is the health promising the molecules which are present in the camel milk. I mean, those who are in camel milk, they have better health than those who are on the cow milk. So the other is like people are very reluctant to the environment now, because you know many people, they are now avoiding to use the milk, any milk. They are vegans people. They are against to use the milk. I mean, now there is like almond milk, soya milk, banana milk, this milk, this milk. So the camel milk is the best alternative because it is coming from an animal. So then. So we can use the camel milk. The camel is a food security animal in the areas like we are living. I mean, the climate heat regions, the desert, where there is nothing. So only the camel can survive and produce and sustain the productivity in challenging climatic conditions. So because I mean, for this, I usually say that we have to adapt. Like, we are already, the camel world is going to 4 type of production systems. I mean, as a major, we are heading towards the 4 type of production systems. So like one, we have like the high input, high output system, like we have in the camel farm intensification. Then we have there are like many camel farms or the dairy farms. They are emerging near the big cities, especially in Africa, in Kenya, in Jigjiga, in many places. I mean, the people are bringing their lactating camels near the city and collect the milk and sell to the cities. That is also another semi-intensive system. And then we have like in Pakistan, the people, they bring their lactating camel near the roads and they sell their milk on the roads. That is another system. So in these two systems, only female lactating camels are in the herd, not the other animals. And the third is the pastoralist system. I mean, you know that They have all the camels in one herd. They produce the milk for their own families, for their own use. So this is like the detail. I mean, like we need this is like 4-pronged policy or production system. Production system, one is more sustainable, like the nomadic system. There is a commercial, modern, like we have in UAE, in Saudi Arabia. But I will not say this is intensive production system, Dr. Bernard, especially for you. I would like to say this, because in many conferences I have seen the people say this is intensive system. I think this is not intensive system. Intensive system, you should have a specific breed or type of animals, like Holstein Friesian, like for chicken, for hogs, for other things. For camel, we don't have specific breed. We bring, I mean, camel from different regions, from different places. The only attraction is their milk. If they are producing more milk, they are welcome to join our team. Otherwise, no. So there is no specific breed. There are many things which we cannot put this system in intensive production system. So then we have like the family dairy livestock. Like I gave you the example of Pakistan, and then we have semi-intensive lactating herd system like in Africa. So these are like the 4 major systems. And almost, this is my estimation, not very true data, that the sustainable system is producing almost 75%. I mean, the nomadic system, 10% is coming from the modern commercial system, like I told you, our farm, and then 8 to 9% from semi-intensive system, and 6 from the the system like we have in Pakistan. So then we have these are like this is like the best I think the best camel breed for milk. This the name of this breed is Brela. Brela is very good. I mean, we had one camel before we fought. I mean, with many people, I was saying that the camel can produce up to 30-liter milk. So many people, they opposed me. They said no, it is not possible. We had one camel. I still remember her number, 444 in our farm. She produced one day 42 liter. 42 liter. So Brela is the amazing camel. She was a Brela. Amazing camel. Amazing camel. But I'm not in favor to collect all the highest yield camels. There is another issue that we will talk. So they have, I mean, this is the sustainable system, the nomadic system. They have their own power, their own strength, because they know how to survive in this environment. They have their own rich breeds, the types of the camels. They have skills to keep the camels. They are very highly adapted to the, I mean, they have very good types of breeds. The main issue is in some areas, now the peace is the problem, so some people, they are suffering, the camel people, like in Pakistan, Afghanistan, in the border, there is fighting, there is war, now the border is fenced, so the people, they cannot travel with their camels to go in the summer to different areas, in winter to different areas. Like there are issues. I mean, then they have like poor health facilities. They have lesser access to the camel milk market. That is the problem. Therefore, we say we should not have a centralized standard system, because it will lead to kick out the pastoralists from the camel milk business. So then they have poor channelization. They are, I mean, the central part of the team. They are, I mean, they are producing milk in very harsh and climatic conditions, but they should benefit from the camel milk industry. I mean, that is like very important. We have to keep this in mind. And then the semi-intensive, like our modern system, we have like a highly commercialized farm, herringbone milking system. We are milking 42 camels in one time, and we are producing like 100 camels in one parlor. In 1 hour, we milk 100 camels, and we have better health facilities. We have round-the-year milk production, and we produce. This is very important. in this system, in commercial system, we must consider the ethical part of the production system. We should not push the camel to the, to the wall to produce milk and to calculate their nutrient and to just take the milk and give a lot of medication and these things. No, we must go for an ethical system. Otherwise, there will be no difference between the other milk and the camel milk. So then the basic feed is, in our farm, we provide feed 24 hours. The camels are not, calculated that you produce this milk, you will eat this, otherwise no. We are providing the basic feed like the hay 24 hours, the water 24 hours. Then the additional feed, which we call in Arabic khalta, TMR, total mixed ration, that is provided as additional food for their to fulfill their new requirements, especially the energy. So then we have the the same, we should not follow the cow model. I am sorry to say, but that is like the very, and some people, they say the camel cannot produce milk this much, but we have the experience. The camel, they are producing 9 liter is like the target we can achieve. 9 liter per year. Average 9 liter for a lactation of 11 month or 10 days less than 11 months. So you can see the camel, they are producing milk and they are they reach to 20 liter, 21 liter. This is like normal of one house. You can see they produce like 20 liter, 15 liter, 18 liter or something. So then we have like, here you can see the daily yield, days in milk, and then we have the, there is one other question which is very important, the AMT, actual milking time, because some camels, they have really very hard system. It is very hard to milk them by machine. So because camel teats are not homogenized, their udder is different. In one camel, the teats are different. One teat is longer, one is wider, one is, thick, one is thin. So there are issues. But still, we say that if the actual milking time is from 3 minutes to 5 minutes, this is the best camel. Because when they are milking short time, they have better stress control. They are happy camels. So we need more milk, yes, but at what cost? We must consider more health challenges will be there if we will increase the milk, because we have the experience. When the camels which are producing more milk, they have more health challenges. Like their fertility goes down. If you will see the record, the camels who produce more milk, they have less replacement in the farm. They have less calves in the farm. So then the milk driven policy is also in this system. It is like very complicated thing. Please, you understand what is the milk driven policy. If you just milk the camels, and if you milk and milk, and they will not go to the breeding. So the good camel will be keeping out of the breeding system. Because some farms, even in our farm, before our policy was the camel which are producing more than 5 liter, they will not go to breeding. So there were some camels, they were producing for more than 1 year, they produce more than 5 liter. So they did not come in the breeding system. So these are like the very important points. And then the average, this is my dream camel. If a camel can produce 10 liter milk per day average with a lactation period of 10 months, this is like our dream target. Actual milking time, we in Arabic, I don't know what you use the word, therefore there we use the word fashush. fashush camel, I mean the easy milking camel, if she is producing milk in actual milking times from 3 to 5 minutes, that is like the best camel. So we have 2 herringbone camel milking parlors. So almost 100 camel are milk in 1 hour from one parlor. So then the calving interval, even if the calving interval is 3 years, this is manageable. But in high yielding camels, this length is even more. I mean, these are the challenges in the intensive farming system or the semi-intensive modern farming. So the community-based, like I told you already, I mean, this is the system which is mostly in Africa near the cities. They have like best community support, strongest sustainable community. They bring the lactating camels to this system. They give very lesser inputs to the system, but they are producing very good quality and more milk. The main issue is now the grazing challenges. Even in Pakistan, we have like camel areas, camel regions. Now those lands are either occupied by the influential people. They started farming there. The natural parks, the national parks, all the government big projects come on those lands. So the camel people are kicking out from the grazing system. So that is like their big challenges. And the overall security, the poor health, we already discussed this area. So this is like very important talk. We must, I mean, wherever we sit and talk about the camel milk, we must discuss this point. We should, we should produce milk for the local market. We should convince the local people to use the camel milk, or we should export, only look for the export. Like I will give you one example. We are exporting camel milk to China, but we are importing Pepsi, Coca-Cola, Red Bull, these things. Why? We should use, I mean, we should connect our next generation with the camel milk, because the camel milk is the real food. The real food. We like, there is now a lot of science on the pellet memory. When you use the ultra processed food, the junk food, I mean, your memory need always such a type of food. Why not we connect our next generation with the camel milk? So then, yes, like I have already said that Red Bull, Pepsi, and we import this and we export camel milk. It is like not, I don't think if it is the right work. So the government should buy the camel milk. I mean, that is like very easy for the governments to buy the camel milk and use it, provide it in the schools, in the universities, in the hospitals. So it will be an additional support to the health, and it will be a very wise policy to connect the next generation with the camel milk. Otherwise, they are forgetting the taste and the flavor of the camel milk. The new generation, they don't want to take the camel milk. They like to have Pepsi, not the camel milk. So if there will be no, I mean, we have to work in this area. That is very important. And the nomad system is already going on very well. I mean, somehow we discussed this area already. Now, this is very important part of my talk, which model to follow. We have to make our own model. We have to give the world our own model, camel milk model, or we have to follow the cow model. We have to decide. We have to decide. And in such platform like we have here, very influential people, they are almost everywhere. They talk about the camel milk. We must consider this point like the very important part of the camel milk, because the policy will lead us to somewhere. I mean, So therefore, dairy cow model is based on specialized breed. We don't have very specialized breed for camel milk. We have some breeds. They are producing more milk than others. Like I told, Brela milk breed, like in Central Asia, Arwana. We have Harani breed in Pakistan. We have Majahim in Saudi Arabia. We have Khawara in Oman and this region in Rub al Khali. But we don't have specialized breed, so we cannot, copy that model. That is number one. Number two, the camel need to eat from a diversity of vegetation. If you will give very specific feed to the camels, you don't know if you will fulfill the requirements of the camels, because they need not only energy source, not metabolizable energy, not only protein, they need some minerals. Very special minerals. We don't have very precise very comprehensive understanding of about the camel nutrition. Exactly, precisely. And that is then the mechanization. If we should go for mechanization and AI, artificial intelligence, robotic milking like in cow, or not, more ethical issues like slurry, wet environment, environmental cost, higher emission. If these are like stressful environment, and also short reproductive life. The camels outside with the nomads, they have like 10 parities. They calve 10 times in their lifetime. In farm, they cannot produce up to 8 times. So that is the challenge. That is the challenge. This way we will erode the very good genetics of the camel. So then we have in the very intensive farm, there is lameness, same like cows, mastitis, metritis. There are many health issues. Then we have the standardization issue. Before, there was few days before Dr. Bernard Fay was there was a conference or seminar about the standardization of the camel milk. The camel milk, there are like two main entities of the camel milk. One is Bactrian milk, which we usually we ignore. They are producing more fats, up to 4 or 5%. And then we have the dromedary camel. They are producing from 1.5 to 5%. We have the dromedary camel, they are producing up to 4% fats. So nobody can impose that the camel should produce this much fats or not. For the other things like total bacterial count, somatic cell count, the other things, yes, these are debatable. We can go for this, but for the composition, I am not convinced to impose some standards on the composition of the camel milk. So this way the pastoralist will be kicked out from the system for sure, because their milk have no homogeneity. They produce, 10 people produce different milk from the other 10 people. So if there will be standardization, I'm sure, because we have this experience with the standardization. Al Marouj, there is Omani company. They are collecting camel milk in Salalah, Oman. They always refuse to buy the camel milk from many people. They told me, the camel farmers told me that they said your fat percentage is less than 2.5%. So just they were kicked out from the system. They are producing milk, but their milk has no value. For the, we should, I don't say follow me. What I say, that is correct. But I say there must be a debate. There must be a discussion. This must be the part of the agenda of this conference also. So, this is like not very important here. I think this was the scoring about the camel. This we can incorporate in the camel industry. So how to make the scoring to select the dairy camels. because we should know what type of camel we are looking for, like the daily average, the lactation yield, the actual milking time, like I told you, milking ability, health model. Some camels are stress-free and some have very, like, especially some camels, they are not happy with the milking. They know big voice, ba, 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 and make the problem very difficult to be milked. So then the calving interval, then the average milk production, and again this fat issue or protein issue, lactose issue. Some people you might have seen when they claim that the camel milk is good, they say there is no lactose in the camel milk, but there is 4% lactose in the camel milk. I get the data every day. There is 4% lactose in the camel milk. There is cholesterol in camel milk. There are fats in camel milk. It is, I mean, different issue. So then the conclusion. One, that the camel has the tremendous potential to produce milk. There is no doubt at all. So the quality milk and the quantity both are ensured. The camel is a traditional breed, need to be understood better. Not, I mean, we can, that is like it must not be treated as a factory breed. It is a traditional breed. In factory breed, we just give antibiotics to kill all the other players and just to give one player the chance to win. But in the camel world, we have to survive in symbiotic relationship with other players. So it must not be treated as a factory breed. While farming, the camel well-being and welfare must be considered, because the camel need good walking, the camel need dry place, the camel need neat environment. So these are like very important things. And the camel milk must be considered as a natural pharmacy. I mean, we cannot compete in the value, because the cow is producing now, the best cow, I think she is producing 100-liter milk. So how your 9-liter milk camel will compete? So we cannot compete. This is not our playground to talk about the quantity. Our playground is the natural pharmacy, the quality of the camel milk. And then I mean, we must support the grazing system, like the phytochemicals are very important. I mean, must be considered as a precious native traditional food item, like as a niche product, and consumption should go to the local people in school, in hospitals. There is an utmost need of time to extend the cause of the camel milk 2024, this conference, and make a clear guidelines for the camel milk production and advocacy. What type of milk we want to tell the people, that what type of milk is this? I mean, the camel milk, from which system it is coming. I mean, that is there must be very clear understanding about the camel milk. Thank you very much. So here.
Thank you, Dr. Abdulaziz. And now we will open the floor for some questions.
Should I stay here or? Okay. Yeah.
الرئيس ولا يعطي الأوام؟ رئيس واحد.
Yes.
Thank you, Doctor. الله يعطيك العافية إن شاء الله. You said the camel milk is a natural pharmacy. You have said in your talk the camel milk is a natural pharmacy. I agree with you. Camel milk, it has a potential healthy claims, but to confirm that, we have to be very, very, very careful of that. It has to be confirmed with too many clinical studies. We cannot say camel milk, it has so and so and so. We know there are too many articles in the literature, but to confirm that and to say it is a natural pharmacy, it has to be confirmed very, 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 very precisely, especially with human studies, clinical studies. Even so, we have to do it with different population. It means probably it works with some population, but it doesn't work with other population. So we have too many confounders going through this pathway to say camel milk is natural pharmacy. Thank you very much.
Thank
So
you.
you
Another
want to, Bernard, you want to add something or should I reply?
You are not here yesterday, but I say exactly that I say Omar Alaj here. That for me, we cannot say that the camel is a pharmacy. This is a counter-sounds. There is his claim, camel milk could be good for the health when the consumer consume it, but you cannot say it's a pharmacy. A pharmacy is a specific concept with medicine, with specific effect on the human health. You cannot cure cancer. You cannot cure diabetes with camel milk. You can help the people. You know, even myself, I'm against the world. It is anti-diabetic effect. No, camel milk has no anti-diabetic effect. Camel milk has a big effect for regulation of the glycemia. It has a big effect to decline the demand in insulin in human body for the diabetes of type 1. It means if you take camel milk, you will not cure your diabetes. You can help the people. You cannot cure cancer. You cannot say to the people that have cancer, please stop chemotherapy, and like this you will be cured with camel milk. No, and myself, I will say for each conference, I say I'm not agree to say that camel milk is a pharmacy. I'm not agree to say that a camel milk is a therapeutic. It's not a therapeutic. It's health effect. It's health claim. It's very important. It's very important for the consumer. But you cannot cure the people with that. And sorry, Razik, but you must be careful with that because a lot of people every month say people they call me, my brother, my father, my mother, I don't know, they have cancer, I want to have camel milk. I say, okay, if you find camel milk is very good for the health, it will help, I'm sure, but don't believe that it will cure. And when you say camel milk is a pharmacy, I think you're not. For me, it's not a help for the producer.
Okay. Sorry. Thank you. No, sorry. No, sorry. I am not. It is working. Hello? Okay. So let we get some, calm, to be calm. What I say, I am standing with my claims. I will not change my claim. I am 100% sure that the camel milk helps. Pharmacy, natural pharmacy doesn't mean it is like the pharmacy level of understanding. The pharmacy, they say this is NSAID, non-steroid anti-inflammatory drug. But the camel is not exactly the non-inflammatory non-steroid drug. But it affects, 1 minute. When there was no science, the people were not living. The science is to dig out the reality. If you have not reached to the reality, it doesn't mean that it does not have the reality. It is your problem. I claim that this water is wine. You have to prove that this is not wine. You cannot say me that I should not say that this is wine. The camel milk really works. But pharmacy in modern system, like I gave you the NSAID drug, the other drug, but it works as a overall revitalization of the health. You know why I started my work on camel milk? I was unable to walk and to go to the washroom. I had like severe rheumatoid fever. Believe me, I took 1,000 antibiotics. I took 1,000 non-steroid anti-inflammatory drugs. Then an old man in our community, he told me to go to those people and take camel milk, live with them, you will get well. By God, in that area, I climbed the highest peak of the mountain. the camel milk work. But now I don't have the data. How did it heal me? I don't say, we will tell the people, you have cancer, you start camel milk. You have diabetes, no. It will 1000% I am sure that it will revitalize your health. It will give you some energy, something. Yes, like I am agree that it work in some area, in some area not. That is different issue, because in some areas they have deficiency for something which is fulfilled by the camel milk. Maybe in the other area they don't have this deficiency. So I don't claim that you put this in the shelf of pharmacy and write on it that this is for diabetes, this is for cancer, this is for... But I am 100% sure that it is revitalizing our health. We have to find. I don't claim. You know, there are six people around my home. Six people. Please listen, this is very important. Very important, I claim. 6 family, but not 5, they are around my home in UAE.
Dr. Abduraziq.
One has severe autism.
Dr. Abduraziq.
Yeah.
We agree, I think all of us agree that camel milk has some very interesting functional properties.
Yes.
But we need to do more science.
Yes.
Based on evidence to claim that it is a drug against particular disease. Okay, so we move to the next question, please. Say Mohamed.
I will need some water.
Yeah. Thank you.
Your people made me emotional.
Go need more and more.
Perhaps some camel milk is better, Dr. Abduraziq.
I have camel milk with me. I have brought. Don't worry.
I will let him drink before.
Yes, please.
I think I would agree with colleagues that we need really scientific data confirming. We need really scientific data confirming this. There is an example that's saying that the best doctor is solving the life of 1/3 of his patients, because the 2 other third, they will be in good situation even if they don't take anything. And that, I think, because we can see the impact of what we are saying, Razi. A lot of people are paying a lot of money to have this milk, 10 times cow's milk. We should have scientific data, comparing people that are drinking camel milk and drinking cow milk or goat milk, and have those scientific data. We cannot advance this because we put people, like said by Bernard, a lot of people are asking me for camel milk. They pay 10 euros a liter because they believe that it is a pharmacy. It is not a pharmacy for me. Right now, perhaps
natural pharmacy. There is very big difference in this terminology.
It could be, but what about cow milk? What about goat milk? We should have comparative studies and have scientific data. And I think it is important because there is an arnak, it's the same in English, for a lot of patients that are paying very, very high costly camel milk as a pharmacy. And I think what we can recommend in this situation is to encourage scientists to provide more reliable data and scientific data for that.
Thank you. Our colleague, please. And we will close with another more question.
صباح الخير جميعا. نشكرك دكتور على العرض القيم. أنا عندي سؤال واحد واللي هو شائع عندنا في ليبيا بين الناس إن حليب الناقة يساعد في زيادة المناعة عند المصابين بأمراض السرطان على أن يكون خام يعني ويشرب مباشرة بعد حلبه، وهذا اعتقاد يعني شائع. فهل مرت عليك مثل هذه الملاحظة أو لا؟ وشكرا.
هذا مجال للدراسات نتصور.
Again, the same question. what to say? I mean, the climate is a little bit very challenging. You know, if you know that the camel is healthy, the raw milk is not big problem. If the camel, you have the screening of the camels, there is no brucella, there is no tuberculosis, there is no paratyphoid, something like that. So like the people, they have their own camel, they use fresh camel milk. Many people I know. I take every morning in coffee, I take fresh camel milk. I do not use pasteurized. But there are many references that if you will go beyond like 80 Celsius, the molecules, the biomolecules, which have some properties, they are denatured. So if it is boiled like our traditional system, when we boil the camel milk, so it really impacts its quality very badly, very negatively. But it does not mean that we promote or educate the raw milk. Raw milk, if you don't know the health of the camel, it is very dangerous. And if someone bring it to you that this is raw milk, you don't know from what type of camel and what is the time between the milking and the raw milk or the fresh milk. When you see and it is fresh from the camel, it is good. But if you don't know the source, I don't support this idea to use the raw milk.
Please, one more question, not on the biological effects of camel milk, but on the environmental issue, development issue, because this is the main topic of this conference. Please. One more question.
One more question, please. So I think if we need to prove that the animal is a model for treating of human disease, we have to do what's called transgenic animals. So the question is, do we have studies doing transgenic camels? So this is the point. So if you need to do, like to put some medication in the milk, or you have to prove that the milk is effective for treatment, so you have to play around that, you have to do that. But we cannot claim that camel, it can treat the disease or, so this is the question.
I understood from the first, when you started, I understood the whole question. There was no need to explain, I didn't that much the whole question.
One question on the...
This is like, you know, I'm sorry to say, I mean, this is even not the question. Yeah? Transgenic animal and this thing, again, depending on the sophisticated technologies and this thing, the camel is itself very specialized biological model. We have to understand. We have to understand better. Okay.
Okay. Please, one question, the last one on the environmental issue.
I am very happy that there are many questions. Very happy.
Yes.
But we can discuss later on also because there are many other presentations.
Please, the microphone to the colleague which is at the right side of the room. We'll end with this question. I am sorry, but we have a time challenge.
So, good morning, everybody. Thank you for a very good and attractive presentation. But really, I feel a little bit confused as not specialist, because based on your presentation and based on what I have seen since yesterday, I cannot highlight it take-home message. So we are talking about sustainability, but half of your presentation, you are talking how we can improve productivity on meat and milk. Mm-hmm. So what I can understood, so we are going for intensification. So we end up with you, professor, you cannot sleep anymore with your camel. If we can go for the other side, we say that so we can present milk as a natural pharmacy. So the price will goes up, and the poor cannot anymore buy and drink milk, and they will sell their milk and buy Red Bull, as you said. So we will stay in the same position. And thank you.
I think I said that one thing we must understand that we cannot, bound the people to work like this, keep the camel like this. They play with the external inputs. If there is demand, they will keep the camels, otherwise not. Pakistan and India, they have sharing one desert. In one area which is in Pakistan, there the camel price is 100,000 Pakistani rupees. Or in the other side in India, there is no half price is not available because there is no demand. So the demand actually decide the system. So we are producing milk, in intensive farming, that really works, but the prices go up and up. And we, like in UAE, our camel milk is 15 dirham per kilo, which is very expensive. Yes, you are right that the poor people cannot buy this milk. That is true. But this system works because we earn money. Nobody can stop this system. So the pastoralists, they are producing in cheaper price, so they have the right to consume the milk. They have the first priority. Yes, the sustainability, if there will be high demand for milk and they will sell the milk, how they will keep the system? They will be deprived of this precious milk. Yes, that is the question. And that is for many other food items in the world. That is another policy issue. You are right, but I don't have exact answer how to settle this issue. This is the issue. This is the very correct question. Thank you.
Okay. Thank you, Dr. Abdelaziz. I will just conclude because I think that we have time for more discussions. ما نحاول بصفة موجزة. أي.
شكرا سيدي الكريم بما تفضلت له. أنا نحب أن نوجه تحية شكر للأستاذ اللي هو قدم هذه الأطروحة، الخاطر أنا نجلبوا لي نظر زوج حالات. الحالة الأولى هي فعلا صيدلية، أنا أوافقك في هذا وأنها صيدلية، الخاطر وقت ترجعوا للأصل، للرجوع إلى الأصل فضيلة. كلما صارت الخبراء أو التحليل أو البحث العلمي، اليوم أنا شخصيا أعتقد كمربي يعني منتج لهذه كلمات الحليب، هو أن ما وصلناش حتى 0.01% من كل الجوانب. علاش؟ اليوم تذكروا في التاريخ السابق، اليوم كل ما هو قريب يشرب حليب السخون بتاع الناقة يبرى. صرت لنا لدغه حشاكم، سامحوني في العباره، وان يعني كل السموم تعرفوها في الصحراء. وذكر الاخ فهمت ولا الاستاذ وان فما هنالك راس روحا، فعلا هذه بتجارب حقيقيه، وانا معناها التمس معناها بكل لطف وان نحط على ايديك هذه هو حقائق دراسات ثابته. أنا أعتقد أن هي فعلا فارماسي متنقلة، صيدلية متنقلة. لهذا الشأن أنا اليوم نأكد وأن كل هذا البحث أنا نحط عليه أيديكم من بحث علمي وطني وعالمي، بكون راكم وصلتوا حتى نتائج طيبة، ولكن راكم في بداية الطريق. اليوم وقت نتحدثوا على الناقة لها مجالات كبيرة في التغذية، وخاصة أذكر الحليب. اليوم تذكروا معناها في السنوات العصور اللي سبقتنا في التاريخ، الشر حشاكم وعافانا وعافاكم الله، عندكم الشر وعندكم يعني وقت اللي يبدا الانسان جيعان يشرب الحليب يروى. في الامراض ما كانتش الادويه، كانت الناس بعاداتها التقليديه. اليوم احنا نرتقوا من فلاحه تقليديه الى فلاحه عصريه، ولذلك انا نحب نثمن مجهودات هذه الدراسه. انا حقيقه اثلجت القلب ومشكور جدا على هذه المداخله، والشكر والسلام عليكم.
شكرا.
قبل الختام عزيز، قبل الختام بعد استراحة القهوة هناك بعض الإجابات العلمية اللي تقدمها زميلتنا في مخبر تربية الماشية والحياة البرية، ولهذا أدعوكم إلى الرجوع بسرعة بعد استراحة القهوة. شكرا.
مازلت استراحة.
بعد استراحة نحكي بعد استراحة.
مازلت. The coffee break is in 1 hour time. But just to channelize the next discussion, I will try just to point out, to emphasize some important issues which concerning the development policies. I think that we have to link them closely to the improvement of livelihoods of local communities. And we have to look for the right indicators to evaluate the real impact of policies focused on the development of camel rearing. We have also to not forget some key words: water efficiency, resilience to heat stress, environmental or ecological environmental footprint, and the issue of animal well-being. And having said that, we will start now with the presentation of the short communications. And we will open the short communication session by the presentation of Dr. Adil Mouad from Fourth Valley State University, USA, on the oocyst cryopreservation. Please.
Good morning, everybody. Thank you for the hospitality. It's a really very, very nice meeting. Okay, and I really appreciate the invitation to be here. So I am Adil Mouad. I am a professor in Fort Valley State University, Fort Valley, Georgia, USA. And I am also a professor at Cairo University in Egypt. And this work done with collaboration of my colleagues in Egypt about oocyte cryopreservation and lesson learned from dromedary camels. It's not working. ما بيشتغلش يا أختي. So this is a little bit of my background. I graduated from Faculty of Veterinary Medicine, Cairo University, and I did my master on camel-assisted reproductive technology in the same school, and then moved to England for PhD. I did my PhD with a professor who did Dolly the sheep, the first cloned animal, Keith Campbell, and then I did a couple of postdoc in Montreal, McGill University, and in Colorado State University. And now I am in Fort Valley, as I said. Fort Valley is south of USA. It's all in Georgia. So the agenda of my talk, I will start with my introduction, objective, material and method, result, and the conclusion. So according to Food and Agriculture Organization, 20% of livestock are under risk of extinction, and at least one breed is going to extinction every month. In human, every female is born with certain number of primordial follicles or certain number of their eggs, and this number decreases by time until the female reaches the age of menopause. Okay, there is no any eggs or there is no any ovarian reserve of the follicle at the age of menopause. Therefore, finding a way to preserve the egg is very important for preserving the fertility. And as you can see here, let's stop rushing to find a husband and start rushing to freeze our eggs. So egg freezing is very important to preserve fertility in animals and human. What are the applications of oocyte or egg freezing? Genetic preservation, seasonal breeding animals, and all we know that camel are seasonal breeders. Okay, so if you preserve the egg out of the breeding season, you can use the egg in the breeding season, okay? And also species conservation, assisted reproductive technologies, fertility preservation, particularly in human. If you know that if the lady is suffering from cancer, it will affect their ovarian reserve. Therefore, some ladies want to conceive later or to have a child in the future. So freezing eggs is very important for those ladies. Fertility preservation, oocyte banking and donation, social egg freezing. What are the different techniques for cryopreservation? We have two main techniques, slow freezing and vitrification. Slow freezing is gradual decrease of the temperature from room temperature to liquid nitrogen temperature, which is minus 26 degrees centigrade. Vitrification is a rapid decrease of the temperature or sudden decrease of the temperature. So you're taking the eggs from the room temperature and put them directly in the liquid nitrogen. What will happen in two cases? In a slow freezing, you can see there is a gradual decrease of the temperature. So as you can see here in the middle of the picture, we can see ice crystal formation. And ice crystal is harmful for the egg freezing or harmful for the cells. Okay, but in vitrification, vitrification means that is freezing without ice crystal. It's like glass formation. So you are avoiding the ice crystal formation. The temperature decrease in the vitrification, you can see it's almost minus 200,000 degrees centigrade per minute, while in freezing is only minus 0.3 degrees centigrade per minute. Therefore, vitrification is much more better than slow freezing for egg freezing. To achieve vitrification, you have to decrease the volume of the media, because if you have more media, that means you have more water. You have more chances for ice crystal formation. How can we decrease the size of the media? We usually developed a lot of carriers. It's called the vitrification carriers, like open boulder straw, cryoloop, solid surface vitrification, and the cryotop. All of them has the same idea. So the main idea is to use tiny amount of the media, around one microliter of the media, 1 microliter, because if you have more water, that means you have more chances for ice crystal. So what we can see here, we just put in, let's say in cryoloop, we put the eggs in the film and we directly put it in the liquid nitrogen. In solid surface vitrification is we have a metal cube and we take the oocytes or the eggs, we put it in the media and we put directly over the metal cube and they get vitrified. Not only the techniques is affecting the success rate of cryopreservation of eggs, but also the stage. We have different stages of the egg. For example, in vivo inside the animals, what will happen under the effect of hormones, eggs ovulated as mature, and then fertilization happen in the fallopian tube, and they form embryos. The embryo is transplanted in the uterus to form fetus later on. Okay, but in vitro in the lab, you're collecting egg at what is called G2 stage or germinal vesicle, and the egg has to go what is called in vitro maturation. So you're putting the eggs in the media in the lab under environmental condition to be able to mature and then fertilize and form embryos. The success rate or the outcomes of freezing differ according to the stage of the embryo or eggs. So the highest successful rate is the blastocyst, because what is the blastocyst? The blastocyst is the stage that forms the embryo. Blastocyst is many cells. If you put the blastocyst in the liquid nitrogen, if one cell's died, you still have a lot of other cells that can survive and form embryos. But egg is only one cell. If the cells is died, you lost the egg, okay? So why oocytes is difficult to freeze? Egg is the largest cell in the human body or in animal body. It's almost 120 micron. 80% of it is water. So it's very hard to dehydrate. Dehydrate means that to remove the water from the egg and put the cryoprotectant. That's the molecules that protect the cells against the freezing. What else? Low surface area to volume ratio and plasma membrane of the egg. It's very hard after freezing for the sperm to get inside the egg. What are the consequences? Consequences is we can do what is called the parthenogenetic activation, development of the embryo without sperm. What else? Premature release of cortical granules. It changes in the internal structure of the eggs, which is called cytoskeleton alterations, and zona hardening. Zona is the membrane around the egg. So if you have zona hardening, it's very hard for the sperm to penetrate the egg. Not only the techniques and also the stage, but also the species. There is a differences in the species in terms of cryopreservation of the eggs. As you can see from the tables, mouse and human are the highest cryostable cells among species. Among domestic animals, cows is considered the highest cryostables, and also life of a spring produced from vitrifying eggs in cows. When we go down for the other species, you have equine, porcine, buffalo, and the camelid. Camelid is one of the hardest eggs to freeze. Why? Because if you look at the eggs of the camel, they are filled with what's called lipid or fat droplet. And lipid is highly
it's the enemy of freezing. So it's a highly vulnerable to oxidation and degeneration. So why we use the camel? As we all talk from yesterday and today, camel is very important species in terms of milk production, in terms of meat production, fibers, and also racing and transportation. So camels play a significant role in the economy for millions of people living in the arid area of the world. But what is the problem with camelidae? Okay, reproductive efficiency of camel is low compared to other species like cows. Why is that? Because camel is seasonal breeder. Camel has long gestation period, is almost stay pregnant for 13 months. And also, camel has early embryonic mortality and late onset of puberty, okay? In vitro embryo production in camel is not well developed. To produce embryos in the lab or reproductive technology like embryo transfer, artificial insemination, in vitro embryo production, in vitro fertilization is not well developed in camel. And the reason why, because we don't have many slaughterhouses that slaughtering camel females. We don't have ovaries, okay? Therefore, cryopreservation of oocytes would be an alternative way to overcome the limited availability of oocytes, allowing for improvement of in vitro embryo production in this species. So, what we did, we published quite few papers over the last few years about oocyte cryopreservation and embryo production in camels. What is the aim of those papers? Is to establish a technique for freezing of camel eggs, okay? And we can use these eggs for in vitro embryo production later on. What we found here in those people, in these studies, as you can see here in the graph, we used different cryocarriers like open boiled straw or straws or solid surface vitrification, cryotube, and various vitrification solution. Vitrification solution is the cryoprotectant that we use to protect the egg against freezing damage. It's like when you are freezing semen. What is the cryoprotectant when you put in semen? Like glycerol or dimethyl sulfoxide, DMSO, or ethylene glycol. Those molecules can penetrate the egg and replace the water, but they are not free. They cannot freeze. If you put glycerol in the freezer, it will not solidify. It will stay as a liquid. Okay, it's the same here. So what we found here, you can see the vitrification solution 2, which is 25% ethylene glycol plus 25% DMSO, and cryotube and solid surface vitrification have higher maturation rate, okay? And you can see here the maturation indicated by extrusion of the first polar body, which is half of the chromosomes of the camel extruded as first polar body. When we looked at the developmental competence after in vitro fertilization, we found that both solid surface vitrification and the cryotube as a cryocarrier in combination of VS2, which is 20% ethylene glycol plus 20% DMSO, resulted in higher developmental competence, okay? So these two techniques, or the cryocarrier and vitrification solution, is effective for vitrifying camel eggs, okay? But do we have still problem with egg freezing? Yes. What is the problem? What is the problem with egg freezing? As you can see here, if you expose the gametes to environmental conditions like cryopreservation, temperature, culture media, light, transportation, so the environmental condition can cause what's called oxidative stress. Okay, what is oxidative stress? Oxidative stress is imbalance between the antioxidant capacity and the production of reactive oxygen species, ROS. If you have higher level of reactive oxygen species, you have lower level of antioxidant. That means it will harmfully or negatively affect the fertility and affect the production, embryo production. So what we need to do, you can supplement the media with what's called antioxidant. All of us taking antioxidant. What are the antioxidants? Like melatonin, vitamin C, vitamin E. All of those considered antioxidant, okay? So the antioxidant is protecting the egg against oxidative stress. What else? We have what's called coenzyme Q10. What is coenzyme Q10? Coenzyme Q10 is a lipid-soluble vitamins, okay? It's present in the mitochondria inside your body and the animal body. It acts as antioxidant, okay? It protects the cells against oxidative stress. Previous study showed that supplementation of the media, either the semen media or the egg media, can protect the egg and sperm against freezing damage, okay? However, there is no studies done in camel. So we started to put hypotheses how if we use the coenzyme Q10 in IVM or in vitro maturation and vitrification can affect the developmental competence of egg. And we came up with these objectives. So the objective is to evaluate the effect of CoQ10 supplementation during in vitro maturation and vitrification of dromedary camel eggs. And we also analyzed what's called the antioxidant and oxidative stress in the media. We took the media after IVM and we did a spectrophotometer to see the antioxidant effect of CoQ10. How we did that? So we collected camel ovaries, as you can see here, the camel ovaries from dromedary camel from a slaughterhouse, and then we collected camel eggs from those ovaries. You can see the shape of the camel ovary, okay? Shape of the camel ovary is like strawberry shape, okay? And then the egg is called cumulus oocyte complex. We vitrify them with solid surface vitrification. We mature them after vitrification in the lab. So the camel maturation takes almost 36 hours. It's very long, not like cows. Cows, only 24 hours, the oocyte can be matured. And then we did in vitro fertilization by epididymal sperm. We get testicles from the slaughterhouse. We collect sperm from the epididymis, and we let the embryo develop up to the blastocyst stage for 7 days. And here is the solid surface vitrification. We can see that the red drop on the metal cube is the media within the egg. Okay, so the eggs or oocytes is in the very tiny top on the metal cube. It's only around 1 microliter or 2 microliter, contains around 5 to 10 eggs. We just drop them in the cube and then it becomes vitrified. And you took them by forceps and put in the cryovial and keep them in the liquid nitrogen. You can keep the eggs in the liquid nitrogen for years, like 10 years, 5 years. It's like the sperm. What are the experimental design? So we have 3 experiments. The first experiment, we measured or we tested the 3 different concentration of CoQ10, 2, 25, 50, and 0, 25, 50, and 100 micromolar of CoQ10 in the media. And after that is the best concentration that comes up with the highest developmental competence. We used it in the vitrification solution. So if you see experiment 2, we have 4 groups. Either we didn't supplement the CoQ10 in vitrification and IVM group, or we supplemented only in vitrification media, which C, we supplemented in the IVM media, or we supplemented in both media. In experimental 3, we did measured the antioxidant capacity. And we did a statistical analysis by ANOVA, and we set up the significance at B less than 0.05.
Dr. Mohamed, could you summarize, because the time
I will finish.
You
now.
have used your 15 minutes.
Okay, so our result here, as you can see, 50% micromolar is the highest developmental competence. So as you can see the embryos here, 50 micromolar is the highest developmental competence. And we can see in vitrification, we found that supplementing the vitrification media with 50 micromolar CoQ10 giving the highest survival rate, and this is the oocytes, is damaged oocyte. And again, when we supplemented the CoQ10 into the IVM media, it resulted in the highest developmental competence, and you can see the blastocyst stage here. So, and when we tested the antioxidant capacity, we found that the supplementation of CoQ10 in IVM media significantly decreased the MBDA, which is melanaldehyde, and increased the total antioxidant capacity. So we can conclude that supplementation of CoQ10 during in vitro embryo production in camels support their viability and developmental competence. And among the different concentration, 50 micromolar is the best concentration. And also adding CoQ10 to IVM media and vitrification solution resulted in higher developmental competence. The result assure that CoQ10 is assure the antioxidant capacity or can protect the eggs against the oxidative stress. Okay. We are not only working on camel, but also we are doing a lot of work back in USA in different species like sheep, goat, cows. Okay, we are doing artificial insemination, semen biology. We also funded, my lab is funded by almost $2 million last year from USDA, which is the biggest funding agency in USA for establishing the assisted reproductive technology in different species. So I am happy to collaborate with anyone here. If you want to collaborate with me or with our university, that will be great too, because we are looking for collaboration. We have a collaboration already from Middle East, from Egypt, from other countries, from Africa. So we can set up a collaboration. And here is different people working with. Thank you.
Thank you, Dr. Adel, for this interesting work. And now we move to a second presentation by Dr. Rabah Kelanemer from Algeria, a study of the correlation between follicular size and hormonal kinetics during the postpartum period in the female camel. Is Dr. Rabah in the room? I do not have the impression. So we will go to the next presentation by Professor Bayram Jamali, Genetic Diversity of Tau Maghribi Camel Population in Tunisia. D'accord. So Dr. Tizawi Amal will give this presentation instead of Professor Bayram Jamali. Please, 15 minutes. Try to respect the time.
So good morning, everyone. I'm really honored to be here today among all of you. So I'm gonna present for you my project, my end of year project. So it aims to study milk quality and genetic diversity of Maghribi camel in the south of Tunisia under the supervision of Professor Jamali Bayram and Kamoun Munir. So as an overview, as we all know, camels are of particular interest, demonstrating resilience to harsh climatic conditions. We know also, as we also spoke, that camels are a multi-purpose animal. We use it in meat production, and beast of burden, especially in transport, in sports and entertainment. We use it in hide and hairs, and also it has a very important dairy potential. So here we have the optimum gross composition of camel milk. We see that for fat it's 5.5%, for protein is 4.5%, solid non-fat is 14.3%, water 88.5%. So dromedaries are represented by the Maghribi population, which exists throughout North Africa. It has a significant cultural, nutritional, and economic importance, particularly in Tunisia. They are adapted to arid and semi-arid environments of southern Tunisia and provide a vital source of sustenance for local populations. We can see here the 6 governance in southern Tunisia. The numbers of camels in this map are represented in Gafsa, Tuzurk, Belli, Mednin, and Tatawin and Gebes with 56,000 heads. The milk daily production is 7.5 liters per day and 12,000 tons per year. In fact, milk production capabilities can vary from a population to another due to several facts such as genetic variability. So genetic variability plays a significant role and suggests considerable potential for selective breeding and provide insights into the genetic structure, breed compositions, and relationships between populations and among individuals within each populations. But despite the multiple purposes and socio-economic importance of dromedaries dromedary camels in the south, camel breeding remains less developed and understudied. Their genetic aspects continue to pose challenges and are not fully utilized in agricultural practices. The main three issues in the sector, which is reproduction management, defining criteria for selecting breeding animals, and optimizing the use of camel products and products, which here we can say the value of chain. So this research focuses, first of all, on the milk physiochemical quality, and second of all, on genetic diversity among two populations of Maghrebi camel in southern Tunisia, specifically in the Gafsa region. So the three main objectives is first to evaluate and compare the physiochemical characteristics of milk from two different populations in different regions. Second, to investigate population effect on milk quality variations, and last, to assess and compare the genetic diversity between and within the two populations using molecular markers. So for the materials and methods used in this study, first we start with the milk sampling. The study was conducted, as we said, on two populations of Maghrebi camel in the Gafsa region in the Villa and Luxar. The sampled she camel had an average year of 9 years. We sampled 120 individuals. The data collection was made twice a week and twice a day. The milking, we milked front teats, complete stripping, while the other two teats were reserved for the calf to suckle. To estimate yield from the whole udder, milk quantity was multiplied by 2. For the milk collection analysis, it was collected aseptically during the routine milking in sterilized recipients and transferred immediately to the laboratory for analysis. They were stored in sterile plastic container at 4 degrees Celsius and analyzed to determine chemical composition. We also performed statistical analysis using statistical analysis system SAS software, and the significant difference between means were calculated by one-way analysis of variance, ANOVA test, and the Tukey studentized range test at probability less than 0.05, which highlight the difference between population 1 and 2. For the genetic diversity, the blood sampling was taken in the Gafsa region. First in the Mdilla, we took 52 blood sampling and 68 in the Luxar. The blood sampling was taken randomly, and this is to ensure that individuals are representative of the real population. We also performed a polymerase chain reaction, so the extracted DNA were subjected to the PCR technique, in vitro replication of specific DNA sequences. 9 microsatellite markers were selected to characterize the genetic variability of dromedaries in the Gafsa region, and of course the objective of this step is to allow the generation of billions of copies of a particular DNA fragment from a matrix DNA. Next, the genotyping, which was performed on a computerized sequencer, ABI Prism 3130 applied biosystems using capillary electrophoresis. This software determines the genotype of each sample for each of the 9 studied microsatellites by quantifying fluorescence peaks for each fragment using the size markers. So next we move to the results of this study. First of all, we have this table that shows the milk physiochemical composition between population 1 and 2. We can notice that population 1 is and significantly higher than population 2. For total solid, non-fat, and fat, we can see that population 2 is slightly higher than population 1, but this is a significant difference. Here we can see the protein fraction. We can see that for total protein, casein, whey protein, and non-protein, population 2 is also slightly higher than population 1. Of course, these variations are influenced by camel type and season. So we observed that there is low casein and fat percent. This can lead to two things. First, it decreases the potential nutritional benefits, and second, it affects negatively the processing of camel milk into products like cheese, yogurt, and butter. Of course, this is explained by the limited presence of kappa-casein. The scarcity of kappa-casein leads to the breakdown of the casein network upon cutting, resulting in the loss of the cheese dry matter into the whey. For the statistical analysis results, we noticed, of course, significant statistic difference between both populations for milk yield, ash, fat, whey protein, casein, solid non-fat, total solids, and titrable acidity. And we know for pH, total nitrogen, non-casein nitrogen, non-protein nitrogen, total protein, and density, we noticed non-significant statistic difference between both populations. Moving to the evaluation of DNA quality, we can see here a picture of DNA quality based on agarose gel electrophoresis. We can see a clear singular and intense bands for the majority of samples. We can say that the extracted DNA is with acceptable quality plus suitable as a matrix for the required analysis. Next, here we are evaluating the DNA quantity as so the ratio R indicates the purity of DNA. It should be between 1.8 and 2. So we observe there is a significant quantity of DNA extracted with moderate DNA purity. Next, for the polymorphism rate, we have the intra-population diversity. For the allele ASB17, we have it's the most polymorphic marker, and HMS7 and AHT42, it's the least polymorphic markers. We see that there is a highly pronounced genetic polymorphism with an average of 6 different alleles, so the polymorphism rate is 100%. The total number of alleles observed is 61 different alleles, and an average of allele per locus is 6 alleles per locus. And we can see for Lexar region in population 1, there is 3.2, and for population 2 in the medulla, it's 6.2. So population 1 exhibited higher allelic richness than population 2. This can be explained that we have a less genetic drift, which maintained larger effective population sizes subject to less selective pressure. So the figure below shows the allelic frequencies. We have the distribution of alleles. So for locus 80, it's the most common allele. And for allele 75, locus 75, 76, 77, 84, and 98 is the common allele for population 2, and it's absent in population 1. Here we have the average and total heterozygosity rates per population. We have the expected heterozygosity is higher than observed heterozygosity, which we have a positive deviation, which means there is a deficit in heterozygosity within the population. So for the fixation index, we can see that the HTG6, it's less than 0, so it means that the subpopulation has an excess of heterozygotes. And for ASB17 and HMS1, it's higher than 0, which means that the subpopulation has a deficit of heterozygotes. For the average fixation index, for population 1 and 2 both together, it's an average of 0.358, which is a deficit in heterozygotes. So the results of fixation index confirm those of heterozygosity rates, indicating that the population is not in Hardy-Weinberg equilibrium. This imbalance is explained by two things. First, inbreeding, which alters genotype frequencies and leads to a loss of genetic variability over generations. And second, the presence of new alleles resulting from deletion in the primers, mutations in the flanking sequences of the microsatellites, or alleles that fail to amplify during PCR. Here we have the intra-population diversity. As we see within population, the total genetic variability is 94%, and for between population is 6%. So we observed that there is an absence of geographical and genetic structuring between two populations that suggests they are homogeneous, and there is a close similarity implies that there are exchanges of animals between these two regions. For the gene flow, it ranges between 1.4 for locus VHL20 to 27 for locus HMS1. This indicates a substantial exchange of genes between these two populations. So we can say there is a significant gene flow, and we can say homogenized allele frequency, and it reduces genetic references, which is, so we conclude we have similar populations. So we conclude that the findings from this study underscore the importance of camel milk performance and its nutritious value. Their genetic diversity present within the Maghrebi camel in southern Tunisia. So the assessment of genetic diversity highlights the richness of genetic variants within this specific camel breed. This offers valuable insights for conservation efforts and sustainable management practices, providing a foundation for future breeding strategies aimed at enhancing milk productivity and resilience to climate change. And thank you for your attention. Thank you.
Thank you, Mrs. Tizewi, for your presentation. And we should pass now to the next presentation about association between lipoprotein lipase gene polymorphism and fatty acid composition of camel milk. It will be presented by our colleague in our lab, breeding and wildlife laboratory, Mrs. Latifa Shamkh. We have 15 minutes. Please respect our time to be with on time.
Bonjour. J'ai l'honneur de vous présenter mon travail intitulé « Étude d'association entre les polymorphismes du gène LPL et les caractères laitiers de la chamelle ». Pour introduire, l'élevage camelin en Tunisie occupe une place importante dans les régions arides et semi-arides du centre et du sud tunisien. Il engage 80 000 unités femelles et il compte 80 000 unités femelles et engage 2300 éleveurs. Cependant, et malgré cette importance, la productivité du secteur laitier reste faible. Pour cela, l'exploitation des voies génétiques pour l'amélioration des caractères laitiers est nécessaire sur le plan quantitatif ainsi que sur le plan qualitatif. Surtout sur la qualité de la matière grasse pour satisfaire l'exigence des consommateurs qui sont de plus en plus exigeants, qui sont de plus en plus conscients de la qualité de la matière grasse et l'effet des acides gras sur la santé humaine et qui cherchent un produit avec une meilleure qualité nutritive. C'est-à-dire un lait moins riche en acides gras saturés et moins riche en acides gras trans et plus riche en acides gras polyinsaturés de la famille oméga 3 avec un équilibre avec les acides gras de la famille oméga 6. Les acides gras ont deux principales voies de biosynthèse. Les acides gras à chaîne courte et moyenne sont de nouveau synthétisés dans la glande mammaire et les acides gras à chaîne longue sont prélevés de la circulation sanguine. Ces derniers sont prélevés sous forme de triglycérides, intégrés dans des kilomicrons et des lipoprotéines. Les triglycérides vont être hydrolysés en acides gras libres sous l'action d'une lipoprotéine lipase qui est une enzyme fixée dans les cellules endothéliales et des capillaires sanguins. Ainsi, l'objectif de ce travail est d'identifier des polymorphismes dans le gène LPL qui code l'enzyme lipoprotéine lipase et de rechercher d'associations avec les caractères laitiers, la production laitière, la teneur en matière grasse, la teneur en protéines et la composition en acides gras. Pour atteindre cet objectif, 67 chamelles appartenant à deux systèmes d'élevage, le système semi-intensif et intensif, ont été utilisés. Ces chamelles ont été élevées dans les deux stations expérimentales de l'Institut des régions arides et dans 3 troupeaux privés. Des contrôles et prélèvements laitiers mensuels ont été effectués durant toute la période de lactation de ces chamelles. Pour la production laitière, la teneur en matières grasses et protéines, on a utilisé et analysé les échantillons prélevés depuis le 2e jusqu'à 10e mois de lactation. Pour la composition en acides gras, on a utilisé les échantillons prélevés durant le 4e, 5e et 6e mois de lactation. Pour la teneur en matières grasses et protéines ont été mesurées par la méthode de Gerber et Gjeldal respectivement. Pour la détermination du profil en acides gras, la matière grasse a été extraite selon le protocole établi par Caray et al. 2004. Les esters isométhyliques ont été obtenus selon la méthode standard ISO 2002 et l'identification des acides gras a été réalisée par chromatographie en phase gazeuse couplée à la spectrométrie de masse. Pour la caractérisation des polymorphismes moléculaires dans la population, on a choisi le fragment couvrant l'exon 5 du gène LPL suite à une analyse in silico. L'extraction de l'ADN a été effectuée selon le protocole établi par De Armas et al. 2005. La PCR a été effectuée dans les conditions appropriées de fragments cibles et le génotypage a été effectué par séquençage. Les fréquences alléliques et l'équilibre Hardy-Weinberg ont été mesurées, calculées par le programme GenAX. Les analyses statistiques ont été réalisées par les mesures répétées d'ANOVA à l'aide du logiciel SAS, du programme SAS. Passons aux résultats. La chamelle a produit en moyenne générale 4,21 kg du lait par jour. Les teneurs moyennes en matières grasses et protéines sont respectivement 2,45 et 2,67%. L'analyse des GCMS l'analyse des acides gras par GCMS a permis d'identifier 28 acides gras dont les majoritaires sont l'acide myristique, palmitique, palmitoléique, oléique et stéarique. Le lait de chamelle contient en moyenne générale 59,71 acides gras saturés, 37,14 acides gras monoinsaturés et 3,09% acides gras polyinsaturés. L'amplification par PCR du fragment d'intérêt ou de la région d'intérêt a généré un fragment de 480 paires de bases. Le séquençage a permis d'identifier deux SMP dans l'exon 5 du gène LPL, une substitution de guanine en alanine et une substitution de alanine en guanine. Les deux mutations sont synonymes et ils n'ont pas provoqué un changement dans la séquence protéique. Les fréquences alléliques reflètent l'équilibre Hardy-Weinberg de la population. Par conséquent, les deux SMP sont retenus pour l'étude d'association. Aucune association n'a été enregistrée entre les deux SMP avec la production laitière et les teneurs en matière grasse et protéines. Des associations ont été enregistrées avec, entre les deux SMP avec des acides gras polyinsaturés à chaîne longue. Ceci témoigne le rôle du gène LPL dans la synthèse des acides gras à chaîne longue. L'SMP a affecté la teneur de l'acide C22. Les chamelles homozygotes pour l'allèle G ont produit un lait plus riche en acides gras et l'SMP2 a affecté les teneurs des acides C22, C22A et C22C, qui est un acide appartenant à la famille oméga 3. L'effet de ces mutations suggère qu'elles sont en fort déséquilibre de liaison avec d'autres mutations non synonymes ayant un effet sur ces mêmes caractères laitiers. Pour conclure, les deux SMP identifiés dans l'exon 5 témoignent le rôle du gène LPL dans la synthèse des acides gras à chaîne longue. Ces deux mutations ont un effet sur la qualité du lait de chamelle. Ces résultats offrent des pistes pouvant mener au développement des marqueurs génétiques qui pourraient aider à la sélection des camelins dans le but d'améliorer la qualité du lait de chamelle, mais restent des résultats préliminaires car l'effectif des chamelles est limité pour permettre une analyse statistique valide. Et merci pour votre attention.
Thank you, Latifa, for your clear presentation. We pass now to the next presentation entitled Isolation and Characterization of 1B Genotype Vancomycin Resistance Anticoxin from Camel Milk in the southwest of Algeria. If he's in the room, no. Okay. We should pass to the next for our colleague Samia Hleli from Tuzer. Samia Hleli will give information about comparative study of methane emission produced by camlet and cattle. Thank you.
Merci, bonjour tout le monde. Je suis heureuse d'être parmi vous aujourd'hui pour présenter des résultats préliminaires concernant une étude comparative des émissions de gaz à effet de serre par les camélidés et les bovins dans la région de Tozeur au sud tunisien. Ce travail est encadré par Dr Moaz Zayed et Dr Tahan Najar. Il est fait dans le cadre d'une thèse de doctorat à l'INAT. Donc, on va voir ensemble une petite introduction concernant les gaz à effet de serre, la problématique, le cadre de l'étude, objectif de l'étude, matériel et méthode, résultats, et on va conclure par les résultats trouvés. Donc, pour les changements climatiques, une petite définition, on appelle changement climatique les modifications du climat accompagnées d'une augmentation générale des températures moyennes au niveau mondial. Ces modifications sont dues à l'augmentation de la concentration des gaz à effet de serre qui sont dégagés par plusieurs facteurs qu'on va citer ensemble. Donc, selon la Convention cadre des Nations Unies sur les changements climatiques, ce sont les changements de climat qui sont attribués directement ou indirectement à une activité humaine. Essentiellement, ce sont les activités humaines qui induisent ces changements climatiques en altérant la composition de l'atmosphère mondiale et qui vient s'ajouter à la variabilité naturelle du climat observée au cours des périodes comparables. Donc, il y a, comme je l'ai avancé, il y a plusieurs causes de ces changements climatiques que sont surtout vécus pendant ces dernières années. Citons essentiellement l'influence humaine sur le réchauffement de l'atmosphère, des océans, qui ont entraîné en fait des changements rapides et généralisés. Donc, on peut résumer exactement ces activités dans l'utilisation massive d'énergie fossile, comme le transport, les bâtiments, l'agriculture, l'industrie, transformation et tout. Et puis, essentiellement, la déforestation qui est vécue particulièrement dans les zones arides où il y a une grande déforestation, l'intensification de l'élevage aussi et des cultures et la pollution due aux procédés industriels. Donc, ces changements climatiques ont des impacts indissociables en fait, puisque lorsqu'il y a un déséquilibre quelconque dans l'écosystème, donc ça va entraîner le déséquilibre de toutes les composantes. Ça va bien sûr agir sur notre sécurité alimentaire, sur les animaux, les végétaux, sur l'atmosphère lui-même et tout. Donc, il y a beaucoup d'impacts néfastes qui sont déjà en alerte. Donc, pour la Tunisie, l'impact des changements climatiques est a engendré une augmentation des températures d'une moyenne de 1,1 degré Celsius. Jusqu'à 2030, déjà, on va vivre ces augmentations de température. Il y a pratiquement une différence de température moyenne entre les zones côtières et les zones continentales. Et ça, par exemple, pour Guebs et Tunis, il y a une différence de 1,2 degrés Celsius. Il y a l'accentuation de l'augmentation des températures moyennes qui est prévue jusqu'aux années 2050 et même des études ont montré qu'il y aura des scénarios très, entre guillemets, catastrophiques jusqu'à l'année de 2100. Donc, il y aurait des fréquences augmentation des fréquences et d'intensité des années extrêmes. Hier, on a notre collègue a cité qu'il y avait en Tunisie presque 9 ans successifs ou 9 ans de sécheresse, dont 5 ans sont successifs, et donc ceci a vraiment impacté l'élevage et l'agriculture de façon générale en Tunisie. Donc les périodes plus sèches et humides devraient varier fortement d'une saison à l'autre. D'ailleurs, on peut remarquer, par exemple, en Tunisie, la pluviométrie, elle n'est plus comme avant, même les quantités et la fréquence, elles sont trop changées. Donc, il y aurait aussi baisse modérée donc de ces précipitations. Ici, j'ai voulu vous donner juste une petite idée sur les températures moyennes dans quelques stations de la Tunisie. Et vous voyez voyez en rouge la région de Tozeur, il y a pratiquement une constance au niveau de la moyenne des températures, à peu près 22 degrés Celsius chaque année. Donc, les températures extrêmes basses et hautes sont vraiment très élevées. La moyenne 22 et parfois même 23 degrés. Donc, ça fait vraiment de la peine pour cette région, comme d'autres, bien sûr, surtout sud-tunisiens. Pour la pluviométrie, elle est baissée même jusqu'à 54 et 76 millimètres par an, ce qui est vraiment très grave pour les parcours et qui a engendré justement la dégradation des parcours et par la suite l'obligation à utiliser d'autres modes d'élevage ou bien d'autres produits ou bien d'autres intrants pour substituer ces ressources fourragères. Donc, la problématique, c'est quoi? En même temps, c'est que les changements climatiques impactent lourdement les animaux, les végétations. Et les animaux, en d'autre côté, c'est une relation interactive. De l'autre côté, les élevages sont accusés de causer des changements climatiques et qui ont contribué à presque 14,5% dans les gaz à effet de serre totaux. Et ça, essentiellement par le dégagement du méthane empirique. Donc, la thèse étudiée, c'est la méta-analyse prédiction des émissions de gaz à effet de serre induites par les animaux d'élevage, leur effet sur les changements climatiques et les procédures d'atténuation par la résilience et la préservation de l'environnement. Nos objectifs sont essentiellement deux. C'est premièrement comparer les quantités de gaz à effet de serre émis par les bovins et les camelins au sud-tunisien et ensuite prédire l'intensité d'émissions pour la production laitière et de viande chez ces deux espèces. Nous savons très bien que jusqu'à maintenant, il n'y a pas vraiment, surtout je parle de la Tunisie, il n'y a pas de modèle pour prédire ou de moyens pour prédire ces quantités de gaz à effet de serre. Donc, par contre, partout dans le monde, en France, à l'Europe, de façon générale, Canada, les États-Unis, l'Australie, tous ils font des études sur les gaz à effet de serre, les émissions de gaz à effet de serre, essentiellement le méthane, mais les méthodes de mesure sont très lourdes, très chères. C'est pour cette raison-là qu'on a établi plusieurs modèles, mais qui s'appliquent justement sur la zone où s'est fait l'étude. Donc pour la Tunisie, on n'a pas encore établi de méthode ou de modèle pour prédire ces émissions à effet de serre, ces gaz à effet de serre. Et d'autre côté, c'est que les dromadaires ne sont pas amplement étudiés concernant les gaz à effet de serre. On a quelques études essentiellement en Australie. Il y a, je pense, une ou deux études au Maroc. Je pense une étude en Égypte. Donc, c'est pas vraiment aussi bien étudié. Donc, pour nous, on a essayé de calculer ou d'estimer les gaz à effet de serre émis par les bovins et les camelins à travers l'application GLIM établie par la FAO. Donc, la zone, comme j'ai annoncé cette aux heures, des enquêtes ont été menées dans 5 fermes de bovins laitiers de Ras-Holstein et avec une moyenne de lactation 5356 et 5 fermes de chamelles laitières avec une lactation moyenne de 1800. Ces chamelles sont destinées à la production du lait pour la pasteurisation à 12 heures. Donc, concernant les données, on a fait exactement les collectes des données zootechniques demandées. D'ailleurs, par cette application GLIM, on n'est pas obligé d'instaurer de nouvelles enquêtes. On peut utiliser les enquêtes déjà faites par cette application, qui est une application ouverte à tout le monde, qu'on puisse utiliser pour déterminer ces quantités de gaz à effet de serre. Juste juste introduire les collectes des données zootechniques, que ce soit pour les bovins ou les camelins, comme l'âge de l'animal, l'âge à la première parturition, la production laitière, la composition du lait en matière de matières grasses et matières protéiques, donc la taille du troupeau, la production de viande, concernant la production de viande, bien sûr, l'âge à la naissance, l'âge à l'abattage. Donc, il y a plein d'informations qu'il faut introduire pour faire le calcul de ces émissions. Concernant les paramètres de reproduction essentiellement retenus sont la fertilité, aussi bien la mortalité des jeunes, mortalité des adultes, le taux de remplacement et bien évidemment pour les systèmes utilisés, comme j'ai avancé, nous sommes tous obligés d'adopter presque à tous heures 93% adaptent le mode semi-intensif pour les camelins et bien sûr 100% pour les bovins. Donc, ceci, c'est juste pour vous donner une idée sur l'application. On a utilisé la version 2022. Ce logiciel, donc, pour une idée sur les informations qu'il faut introduire concernant les paramètres de production de l'espèce choisie. Donc, vous trouvez presque toutes les espèces pour évaluer ces émissions. Ici, comme vous voyez, donc les paramètres de production aussi pour les camelins et bovins de boucherie. Pour une enquête bien détaillée concernant les rations utilisées pour ces productions. Donc, pour nous, pour ces deux espèces, comme j'ai avancé, on a choisi des fermes de production laitière et des parcs d'engraissement de bovins à viande. Donc, pour la part des grains dans la ration, on a remarqué ici qu'il y avait presque un tiers de la ration est composée de grains. Donc, comme résultat, la réalité, c'est que cette application, ce logiciel nous donne plein de résultats. Vous pouvez avoir même plus que 50 résultats. Mais ce qui est nécessaire pour nous aujourd'hui, c'est l'étude de ces deux paramètres, l'intensité de l'émission et les quantités. Donc, pour les camelins laitiers, on a une quantité de gaz à effet de serre globale de 1921 par tête par an pour les camelins laitiers et pour les bovins laitiers, c'est à peu près 4431 kg de CO2 équivalent. Par contre, la quantité d'émissions en relation avec la production laitière, elle était de 759 000 pour les troupeaux camelins et 1575 pour les bovins, presque le double des quantités, les quantités dégagées. Comme c'est clair, si selon l'histogramme, le premier, vous voyez que le méthane empirique représente à peu près 75% du méthane des gaz dégagés. La quantité de gaz à effet de serre global émis par ces troupeaux est de l'ordre de 2900, à peu près 3 millions de kilogrammes CO2 équivalent par an. Les quantités de CO2 par tête et par an émis par les bovins à viande, c'est 20 800 et pour les bovins à production laitière, 600 500, alors que pour les camélidés laitiers, c'est 343 000. Donc, c'est très net que les bovins émettent beaucoup plus de gaz à effet de serre, surtout CO2 par rapport à l'autre espèce. Pour le méthane, on a trouvé que le méthane empirique ou le méthane de fermentation qui représente déjà 75% est de l'ordre de 20 000 pour ces deux troupeaux, alors que les bovins laitiers 31 000 et les bovins à viande 1300. Donc en total, vous voyez que pour le gaz, le méthane empirique, il est très important. Pour le méthane provenant de la gestion du fumier, elle est beaucoup moins faible. Pour les camélidés, c'est alors de l'ordre de 280 kilos par an, alors que pour les bovins ensemble, les bovins, tous les bovins, c'est 1800, c'est plutôt 434 000 kilos par an. Pour le total, les camélidés en fait émettent beaucoup moins de méthane total par rapport aux bovins. Ici, c'est juste pour vous donner une idée sur l'intensité des émissions pour les bovins et les camelins, que ce soit pour la production laitière ou la production de viande. Donc l'intensité des émissions des gaz à effet de serre pour produire 1 kg de protéines lait, le Milk Emission Intensity, c'était de l'ordre de 39 kg CO2 équivalent par kilogramme de protéines. Par contre, pour les bovins Holstein qu'on a utilisés, donc c'était 48,7 kg de CO2 équivalent par kilogramme de protéines. Ça, c'est pour le lait. Par contre, pour la viande, c'était beaucoup plus élevé. Pour les dromadaires, l'intensité d'émission est de 72,91 de kg CO2 équivalent par kilogramme de protéines. Par contre, les bovins Holstein, c'était de l'ordre de 91 kg. Donc, c'est bien sûr, c'est plus élevé. L'émission de méthane empirique, c'était pour les dromadaires maghrébis, une moyenne de 0,141 kg de méthane par tête par jour et une émission totale de 52 kg par an. Par contre, les bovins laitiers émettent 0,215 kg par tête par jour et une quantité de méthane empirique totale de 104 kg par an. Donc, pour conclure, les émissions globales de gaz à effet de serre sont plus élevées, à peu près 220% pour les bovins que les camélidés. En moyenne, il y a pour l'émission de méthane, de CO2 et de l'N2O représentent à peu près 70, 28% et 2% respectivement pour les deux espèces. Donc, c'est que ces deux espèces, puisqu'on a dit déjà que les animaux d'élevage dégagent beaucoup plus de méthane, donc c'est clair que c'est 70% de méthane du total de gaz à effet de serre dégagé. L'intensité des émissions pour produire 1 kg de protéines de lait est plus élevée de 123%, alors que pour produire 1 kg de protéines de viande, l'intensité d'émission des bovins est 125% de plus par rapport aux camélidés. Donc, pour conclure, c'est très clair que les bovins émettent plus de gaz à effet de serre et essentiellement de méthane par rapport aux camélidés, aussi bien que l'intensité de production du lait, elle est plus faible, que ce soit pour le lait ou bien pour la production de viande, elle est plus faible pour les camelins que les bovins. Donc, d'ailleurs, le pouvoir de réchauffement varie pour le CH4, il est plus élevé que le CO2. Et en même temps, nous savons que les bovins sont plus émetteurs de gaz à effet de serre, contrairement aux camélidés qui sont connus comme étant moins émetteurs de gaz à effet de serre. Et merci.
Je vous remercie pour votre présentation, qui est en relation avec la question, donc une des questions les plus importantes actuellement, donc le réchauffement climatique. Et maintenant, nous allons demander aux collègues qui ont des questions de bien vouloir intervenir pour poser des questions aux trois présentations qui ont été faites ici.
Merci, Monsieur le Président. Moi, j'ai deux petites questions à la présentation concernant le génotypage, les études génétiques. Madame, juste. Alors, la première question, c'est dans quel laboratoire ont été effectuées les analyses, surtout pour le génotypage, parce que ça n'a pas été signalé dans votre présentation. Et la deuxième question, moi, avec les collègues, on a dans le cadre du suivi des maladies à prion dans la région de Tataouine, on a fait en parallèle des études de génotypage également. Et bon, moi je suis pas spécialiste dans le génotypage, mais les résultats qui ont été donnés parallèlement, on nous dit que c'est le wild type. Wild type. Sauvage, voilà, c'est type sauvage, donc ils n'ont pas trouvé vraiment une... parce que je sais, par exemple, les maladies à prion chez le mouton, par exemple, il y a un gène, enfin, il y a des souches carrément de génétiques différentes et qui sont plus ou moins prédisposées à développer la maladie, etc. Et merci.
Merci beaucoup pour la question. Les analyses ont été réalisées dans le laboratoire de biotechnologie animale à l'École supérieure d'agriculture Mater. Concernant les microsatellites utilisés, on a choisi à partir d'une étude bibliographique des séquences qui sont stables et qui peuvent être potentiellement stables d'une génération à une autre, et ça peut refléter la stabilité ou bien l'équilibre ou bien le déséquilibre par rapport aux conditions de la loi de Hardy-Weinberg. Donc ça a été choisi en fonction de ce qui existe au niveau de la littérature. Pour la deuxième question, qui peut-être d'après ce que j'ai compris, vous avez essayé d'identifier un génotype en relation avec la résistance ou bien la sensibilité à une pathologie. Voilà. Puisque peut-être que s'il y a une variabilité, ça n'était pas touché par les analyses. Et pour les séquences analysées, dans votre cas, vous avez ciblé, je crois, une mutation ponctuelle. Et l'absence de cette mutation ne veut pas dire qu'il y a une association entre les phénotypes observés. Peut-être pour le cas de cette mutation, ça a été identifié à l'échelle internationale, donc ça ne reflète pas réellement le potentiel de nos ressources autochtones qui existent dans le sud tunisien. Peut-être il faut chercher sur d'autres fragments. Et peut-être notre professeur Sela Habib peut.
Non, juste une petite remarque pour le génotypage. Lorsqu'on génotype plusieurs échantillons et on cherche des mutations, on est obligé d'utiliser une séquence qui sera comme référence. C'est les wild type. Donc toutes les séquences qu'on va obtenir, qui éventuellement vont contenir des mutations, on va les comparer avec la séquence des références, les wild type. Donc c'est ça la procédure. Juste, Monsieur le Président, j'ai une question pour Madame Samia Hlali, pour l'émission de gaz à effet de serre chez les camélidés et chez les bovins. Bon, j'ai été un peu étonné par les données qui ont été obtenues concernant la production de gaz par kilogramme du lait. Donc, pour les données, vous avez donné une moyenne de production de 1800 kg de lait pour les camélidés et de l'ordre de 5800, je pense, pour les bovins. Donc, a priori, même si la production de gaz à effet de serre pour les bovins est supérieure aux camélidés, on s'attend à une diminution de cette production par kilogramme de lait. Parce que les bovins produisent 3 fois plus de lait que les camélidés. Donc ça, c'est la première question. La deuxième question, c'est la production de ces gaz par kilogramme de protéines de lait. Pourquoi vous avez choisi les protéines spécifiquement? Donc l'important, je pense que c'est les 1 kg de lait, indépendamment de ses constitutions. Et merci.
Bien, si on peut avoir une réponse, mais très succincte.
Pourquoi on a choisi les protéines? Bon, c'est pas nous qui a choisi, c'est la FAO qui a choisi de prendre en compte les quantités ou la teneur en matière grasse et en matière de protéines dans le lait. Et pour les mesures, c'était en relation avec la production de la quantité de protéines, que ce soit produire de la viande ou bien du lait. C'est l'application de la FAO 2022 qui tient ces paramètres. Sinon, concernant les quantités, elles sont bien sûr plus élevées pour les bovins, malgré que la quantité produite de lait, elle est plus importante pour les camelins. Mais par contre, les camelins, c'est en relation peut-être par rapport au poids vif. D'ailleurs, c'est ce logiciel et la variation de façon générale des émissions de gaz à effet de serre, elle est en relation avec le poids vif, avec les quantités produites et l'alimentation, bien sûr.
Merci. Donc, on prend deux autres questions, c'est Bengoumi et Madame Bad qui est derrière. Je m'excuse, c'est Bengoumi.
Merci beaucoup, monsieur. Donc, j'ai une question commune pour les deux demoiselles ou les deux dames qui ont présenté le travail sur le génétique. Donc, je ne suis pas généticienne, hein, rassurez-vous, mais j'aimerais bien avoir quelques éclaircissements. Je pense que l'expression génétique, elle est liée à d'autres facteurs externes. Si un animal il s'exprime sur son plan, par exemple, productif, donc il peut avoir son potentiel génétique qui va être exprimé en fonction des conditions environnantes, notamment l'alimentation. Est-ce que dans votre protocole expérimental, à toutes les deux, hein, quand vous avez évoqué le lait, surtout le taux de matière grasse et la quantité, et après la génétique, donc c'était quoi la théorie? J'ai pas bien compris. Est-ce que si vous avez oublié de tenir en compte du facteur alimentation, donc je vous assure qu'il y a une lacune dans le protocole expérimental et à mon avis, à mon modeste avis, que les résultats ne sont pas de ce point de vue, hein, de cette théorie-là, ne sont pas fiables, hein, sans compter l'effectif, hein, le N qui était trop petit, c'est-à-dire pour parler d'une mutation ou d'une variabilité génétique. Après, juste une petite remarque pour c'est pas une remarque mais un constat. La comparaison, je pense que la comparaison entre deux systèmes différents pour les émissions de gaz à effet de serre, hein, chère dame, pour les émissions de gaz à effet de serre, je pense c'était un peu difficile de comparer deux animaux qui sont totalement différents, surtout sur le plan des gestions. Donc, puisque c'est ça qui contrôle les émissions des gaz, surtout le méthane, et deux systèmes différents. Donc, si le système il n'est pas identique d'élevage, donc les émissions de gaz à effet de serre par l'animal, déjà c'est pas identique, sans compter que comparer un grand ruminant tel que le bovin et le camelin, c'est un peu sur le plan physiologique des gestions, c'est un peu différent. Donc l'expression et les émissions des méthanes, l'alimentation aussi, elle va agir, et donc un système productif intense et un système à l'extensif, comme vous avez évoqué, donc semi, là, c'est pas le même système. C'est-à-dire, c'est pas le même système. Le semi, il y a une différence entre l'extensif, le semi et l'intensif. Donc, pour moi, sur le plan protocole expérimental, je parle pas sur le plan théorie, donc c'est très difficile de comparer des choses qui sont pas identiques. Sur le plan statistique, même le plan statistique, les logiciels, normalement, il faut être logique. On compare des choses différentes. C'est ça. Pas, si je compare les progestérone ou les testostérones entre hommes et femmes, les femmes ensemble, même âge, même sexe, même alimentation.
Vos idées, condensez votre question.
Ouais, c'est ça. Donc j'essaye d'expliquer. Donc c'est ça. Donc ça c'est un point de vue, c'est pas une question. Ça c'est un point de vue. Donc je vous demande pas une réponse. Ça reste à réfléchir ou à voir après. Mais pour la génétique, je demande des explications ou des détails. Merci.
Peut-être pour expliquer les facteurs de variation, on a choisi la même région, on a ciblé les éleveurs pour minimiser les facteurs de variation et pour rendre le facteur le plus important, c'est le génotype. Donc ça a été tenu en compte concernant l'effet environnement. On a minimisé, oui, l'alimentation elle-même. Non. Presque 80% est expliqué par le génotype.
Voilà, si Bengoumi.
Oui.
Et on termine les questions.
Bon, rapidement, donc je vous remercie pour le travail que vous avez présenté, c'est intéressant, c'est toujours, je vous félicite. Et je voudrais parler du GLIM-I. En fait, GLIM-I, il a été conçu pour des estimations au niveau des pays. Donc, de manière globale, à l'échelle nationale, on peut le faire à l'échelle d'une région, on peut le faire à l'échelle d'une ferme aussi, mais ça reste toujours des estimations. Donc, c'est un travail intéressant. On aurait bien aimé le voir. On peut le sortir aussi par kilogramme de lait, par kilogramme de viande. Ça, c'est sûr. Par contre, là où je ne suis pas d'accord avec vous sur la conclusion. Mettre les bovins de manière générale, à la rigueur spécifiée, la Holstein qui produit 6000 litres dans la région de comme ça, parce que avec les autres races de bovins, on ne sait pas. Donc, dans la conclusion, essayez d'être beaucoup plus précis dans le contexte tel, pour la Holstein qui produit tel, etc. Et pour le camelin, je ne sais pas si on prend les chamelles de monsieur Razik, qu'est-ce qu'on va avoir. Donc, mais c'est un très très bon outil qui est gratuit, hein. Donc il existe en ligne, mais c'est beaucoup plus. On a fait d'ailleurs une formation avec l'OEP, on a des gens formés ici sur le logiciel, qui est gratuit, mais il permet beaucoup plus les estimations au niveau régional, au niveau, on peut le faire même au niveau d'une ferme aussi, hein, parce que on peut justifier les investissements, etc. Mais la conclusion, il faut être assez prudent. Merci.
Bien, je m'excuse pour les autres collègues dont on n'a pas pris les questions, mais on m'informe que le café commence à refroidir. Donc, on vous invite tous à la pause café et on revient à la salle dans 15 minutes. تقليدية